Hello friends, welcome to this lecture series on immune system. This is the first lesson, which would be an introductory lesson, which would give you a very broad sense or broad concepts regarding the immune system. This would be useful for whoever the people who are preparing for NEET PG and also for all the people who are writing for USMLE also. And apart from that, even the doctors who have completed their MBBS and who wants to retain their knowledge, I have prepared these lessons in such a way that these are mainly concept based, and these will give you an immense knowledge on this immune system. Okay, let us go ahead and learn some of the few important concepts in this. So first, we must learn what is a self and a non-self. Self and a non-self. Self system, this immune system first has to recognize the self cells, the cells that belongs to our body, which are working normally, which are good enough to compose in our body. This immune system also delineates the cells which are non-self. That means it can be a microorganism which doesn't belong to our body, or it can be a toxin, or it can be a cell which is malfunctioning within our body, like a cancerous cell. Like there are few exceptions, like commensals are non-self, but body allows it. That concept we'll be discussing later in this lecture series. So primarily we should differentiate between the self and the non-self. This concept we should have a prior in our hand. So there are three defense systems which our body poses against these invaders. So imagine there are so many microorganisms that are attacking in our body. This is the first line defense, that is the barrier system. This is the second line defense, the innate immune system. This is the third line defense, that is the adaptive immune system. The first barriers, like the mechanical barrier, like example is our skin. The skin protects almost most of our body. It protects us from injuries, from the microorganisms, from so many things. Second thing is the chemical barrier. Chemical barrier is something like an acidic juice within our stomach. This is one example. And the biological barrier. Biological barrier is a commensal that is present either in a oral cavity or in the nostrils or in the intestine system. All these things help as a first line defense against the invaders. Then we have innate immune system. Innate immune system is we will be discussing in detail about the innate immune system, but this is the brief overview. This is present intrinsically. It is very non-specific. It won't create any memory, and it has a limited diversity. So this forms a second second line of defense. The third line of defense is adaptive immunity. This adaptive immunity is inducible, and it is very specific. It creates a memory, it is extremely diverse, and it is self-limiting too. So basically our body follows these three, places these three immune defense mechanisms. Let us go ahead and briefly discuss the differences between this innate immune system and the adaptive immune system. See, as I've already told, like look at this picture. So there is John, there is Betty, and there is Matt, and there is Kathleen. All of them in innate immune system is pretty much hardwired. That means it cannot change very much. Whereas the adaptive immune system is varied. You can see different receptors are present for John, different receptors are present for Betty, and different receptors are present for Matt, and different receptors present for Kathleen. Whereas when you observe this innate immune system, all these receptors are similar for all the four people. So that denotes innate immune system is very non-specific. It just goes and attacks. Adaptive immune system is very much specific. It is present intrinsically, which is hardwired, As soon as the invader comes in, it goes and attacks, whereas this adaptive immune system is indivisible. uh, this doesn't create a memory. Innate immune system, adaptive immune system creates a memory. You can see all the receptors here are same, so there is not much of diversity diversity in innate immune system. But when you observe here, the receptors are different for different people depending on the organism they are getting, depending on the immunity they have. So it has a very much high sense of diversity. It doesn't differentiate between the self and the non-self. It just goes and attacks. It differentiates between the self and the non-self. So these are basically different differences between the innate and the adaptive immune systems. Let us go ahead and discuss regarding the innate immune system in brief. Innate immune system performs two functions. One is to directly go and kill the invaders, and then to activate the adaptive response. These are the two functions done by the innate immune systems. This is normally done by the neutrophils, natural killer cells, and then phagocytic like macrophages, dendritic cells, and all. Neutrophils just go and kill the bacteria. The activating, apart from the killing of the microorganisms, activation of the adaptive immunity is done by the especially macrophages and the dendritic cells. These release certain chemical factors which would activate the immune system. Then, how does it does such a function? It has got pattern recognizing peptides or proteins on the cell surface, which would identify the pathogen associated receptor proteins. Receptor patterns. Suppose this is a microorganism, this would have a separate pattern on its cell membrane. This would be identify this pattern recognizing proteins that are present on the cells of the innate immune system. There are various pattern recognizing proteins. Some of them are like toll-like proteins. There are almost 10 to 11 toll-like proteins, and there are also like Mannan binding lectin. So, for example, if you take an endotoxin is a lipopolysaccharide. This lipopolysaccharide would be recognized by toll-like protein 4. And suppose in the cell wall, if there is a Mannan lipopolysaccharide on the cell surface of the microorganism, this would be this lectin, Mannan binding lectin would be binding to this Mannan polysaccharide that is present in the microorganism. These receptors are present over the cell surface. Some of the receptors are present inside the cell surface, like node receptors, or some like RIZ1 helicases. Node receptors, both these receptors present within the cell. Node receptors recognizes the peptidoglycans of the microorganism, and RIZ1 receptor helicase recognizes the nucleic acids present in the viruses. So in such a way, this innate immune system responds, and it also produces TNF alpha, TNF gamma, TNF beta, inflammatory and anti-inflammatory factors, and some of them produce defensins. Defensins will create pores within the microorganisms, Some of them produce interferon alpha, gamma, and beta, where alpha and beta interferons have antiviral property, whereas gamma interferons has pro-inflammatory property. Whereas coming to cell-mediated immunity, cell-mediated immunity functions through B lymphocytes and also through T lymphocytes. B lymphocytes produce plasma cells and antibodies. T lymphocytes act through CD4 and CD8 like cytotoxic T cells and T helper cells, where T helper cells are again divided into T helper cells type 1, T helper cells type 2, and so on. This we'll be discussing in detail when we discuss about the cell-mediated immunity. Okay, this is all about the innate immunity and adaptive immunity. Let us go ahead and discuss about the various lymphoid organs that are present in our body. There are primary lymphoid tissues or primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs, bone marrow is one of them, and also thymus gland, where bone marrow synthesizes the T lymphocytes, which come and they get matured in the thymus gland. And in the secondary lymphoid tissues, we have lymph nodes, and we have spleen and mucosa associated lymphoid tissue called as MALT, mucosa associated lymphoid tissue, like Peyer's patches or tonsils. All these mucosa associated lymphoid tissues comes under this category. Okay, let us go ahead and briefly know about the lymphoid lymph nodes. So here you can observe this is the efferent vessel, this is the efferent vessel, efferent lymphatic, where the lymph is brought through this and the lymph goes out through this. Here we need to differentiate three things like cortex, paracortex, medulla. See, we will be getting stock questions here. So it is clearly differentiated in this figure, where this is the cortex, this region is the cortex region, this is the paracortex region, and this is the medullary region. In most of the cortex region, we find the follicles of B lymphocytes. These are the follicles of B lymphocytes, B cells, where in the paracortex regions, we find the T lymphocytes. In this paracortex region, most of the times we find the T lymphocytes. And in the medullary region where these cords are there, we find the plasma cells, and where there is a medullary sinuses, this medullary sinuses, we find the macrophages. In these lymph nodes, these invaders or the invading microorganisms are presented to this innate immunity. Innate immune cells recognizes them, and then their presence to emulate the adaptive immunity. So if a question is asked like T cells are predominant in paracortex area, B cells are predominant in, P cells are predominant in the cortex area, macrophages are predominant in the medullary sinus, plasma cells are predominant in the medullary cords. And then coming to the spleen, we have a red pulp. See, we have a red pulp and we have a white pulp. In red pulp, this is all is the red pulp. In red pulp, we find the orbices, and then in the white pulp, this is all is the white pulp, and in the white pulp, we find the B cells, and especially when it comes to periarteriolar regions, we find the T lymphocytes. And then apart from this periarterial lymphocytes, we find the B follicles somewhere in the white pulp, and especially macrophages. Macrophages here in the white pulp identify the capsulated or engulf the capsulated microorganisms or bacteria. So that is the reason why whenever there is a splenectomy, that person with splenectomy is prone to infection with capsulated bacteria. This mnemonic is to find out the capsulated bacteria. Even some pretty nasty killers have shiny body. E for Escherichia coli, S for Salmonella. Sorry, it can be this is for Salmonella, or this can be with Streptococcus pneumoniae. P for Pseudomonas, N for Neisseria meningitidis, K for Klebsiella, H for Haemophilus influenza, Streptococcus B series, B for Streptococcus B. Whenever there is a splenectomy, we might have infections with this capsulated bacteria. Having done this part, let us solve some MCQs such that we will be confident on what we are learning. So here is the first question: which one of the following is an attribute of innate rather than the adaptive immune system arm of our host defenses? It is highly specific. Highly specific is adaptive immunity, response to viruses and fungi, but not bacteria. It is again adaptive immunity. Exhibits memory is adaptive immunity. Is a part of our host defense against bacteria, but not against fungi. No, it acts as against the fungi also. Is as effective as the first time it is exposed to the bacteria as it is subsequent times. So every time it doesn't show any diversity, it doesn't memorize. So answer is E. So this is the best answer. Second question regarding antibody mediated immunity and cell mediated immunity. Which one of the following is the most accurate? Antibody mediated immunity, this we would get in the next lessons. This is not correct. Antibody mediated immunity acts in anaphylactic shock. Graft rejection is most associated with cell mediated immunity. Protects against, no, it causes anaphylactic shock. Antibody mediated immunity protects against autoimmune diseases. No. Cell mediated immunity neutralizes extracellular viruses. No, intracellular viruses. Cell mediated immunity protects against fungal infection. So the right answer is E. It protects against the fungal infection. Third question now. Certain components of our immune system are characterized by two attributes: being able to respond specifically to microbes and exhibit memory of having responded to a particular microbe previously. Which one of the following has both? See, it is specific and it exhibits memory. So it should come under adaptive immunity. So adaptive immunity is done by which cell? B cell, basophil, dendritic cell, macrophages, neutrophils. These all comes under innate immunity: macrophages, neutrophils, dendritic cells, basophils, myeloid series cells. So the answer which creates memory is B cell through antibodies and all. And it is very specific. So the answer is B cell. The next question, fourth question. Immune recognition of molecules belongs to self is important to activate natural killer cells, determine the safety of interacting, induce somatic degeneration of B and T lymphocytes, stimulates binding of pattern recognition receptors, triggers an attack of cell expressing the self molecule. This cannot be the answer. Pattern recognition receptors, more than this activation of somatic generation of B or T lymphocytes or pattern recognition, this is to as the safety of interacting with the molecule, whether we are interacting with our self antigens or with the non-self antigens. So the best answer is B. So B is the right answer. Going on to the next question, fifth question. Natural killer cells assess whether other cells are abnormal by detecting types and levels of surface associated. So natural killer cells comes under the innate immunity. They recognize which of these things they have. Natural killer cells have pattern recognition receptors. These pattern recognition receptors would recognize pathogen associated molecular patterns. So MHC class 1 molecules, all this comes under adaptive immunity. So the right answer is pathogen associated molecular proteins. I've already discussed these things like toll receptors, mannan binding lectins, node receptors, RIG1 receptors, which detect the peptidoglycans and viral helicases and all. Sorry, I think I missed a point here. This is the natural killer cell. Actually, this pattern recognition protein receptors would detect the pathogen associated molecular patterns. This belongs to macrophages. But natural killer cells would identify the MSC class 1 molecule. So the best answer would be the A answer. So not this one. This one is the answer. I think we have covered all the introductory topics in this lecture. Thank you for watching this lecture series. Do subscribe to this channel to know the concepts well. Hope it was useful and interesting. Please do subscribe. Thank you all.